gases, I want to spend doing this stuff, which is really the, the heart of respiration or respiratory physiology. How we handle these gases. And we can consider 4 different stages in the handling of gases. Ventilation, which we've talked about. Gas exchange in the lungs. Then you've got to move the gases around the body, and you've got to exchange them in the tissues. So the next 2 classes on these issues. And it's okay, tricky bits. Next class is perhaps the trickiest. So this is what's going on then in the lungs. The gas exchange occurs between the alveolar airspace and the blood, which comes in deoxygenated, and then the gas exchange here, oxygen from the air into the blood, carbon dioxide from the blood into the airspace. And the gases have to cross the respiratory membrane here, which we'll take a closer look at in a minute, but you'll remember what it consists of. The type 1 alveolar epithelial cells here, the basement membranes between these two, these extracellular layers outside the cells, and then across the cell line in the blood capillary or the endothelial cell of the blood capillary, and then into the blood. And all of these gases move in solution. They don't move as bubbles, they move dissolved in fluids. There's fluid on the surface of the alveolar cell, so it dissolves in the fluid, moves across in the cytosol, and then into the plasma, which is obviously fluid. And these movements of gases are governed by the physics of gas movement. I used to hate these gas laws. You hate these gas laws? Yeah. Physics? Well, we can just have a look at them because it makes physics and chemistry relevant to biology. Dalton's law. John Dalton was an English chemist working end of the 1700s, beginning of the 1800s, so he developed this around the year 1800, and he messed around with atoms and gases and molecules, and he formulated this law, which says that, and this is an important one for us, in a gas mixture, let's think about the atmosphere. The atmosphere is a gas mixture. In a gas mixture, the pressure exerted by each gas is independent of the pressures of the other gases in the mixture. And in a mixture, the total pressure of all the dissolved gases is the sum of the partial pressures. Dalton introduced the concept of partial pressures, and that's what we will talk about, because we're talking about a mixture of gases. And each gas exerts its own pressure, and the diffusion of the gas across the membrane is dependent on the partial pressure on one side and the partial pressure on the other side. And since we're talking about diffusion, simple diffusion, the gas will always go from where the partial pressure is high to where the partial pressure is low. It's diffusion. It can't go in any other direction from where it's high to where it's low. So that's Dalton's law, and it's important for us. And we're going to continue on today and talk about this issue of partial pressures and what they are. I'm sorry about my microphone's ringing. And how they are and how they're relevant to respiration. Let me turn this down a bit. A couple of other laws. The ideal gas law. PV equals nRT, which says the pressure of a gas is proportional to its temperature and the number of moles per volume. In other words, the pressure of the gas is proportional to the temperature and its concentration. And you might say, well, how the hell is that relevant? Well, it is. The temperature issue is relevant for us. And I'll point that out. actually in the next class, I'll point out how the temperature issue is relevant for the movement of our respiratory gases. So just underline temperature and put it on the back burner till next class. And finally, Henry's law. William Henry was also an English chemist working at about the same time as Dalton, and he was messing around with gases. And he developed this law which says that the volume of gas absorbed by a liquid is proportional to the pressure of the gas. Important for us. If you drink a lot of Coke, you know this law instantly, because you bring the Coke bottle home, you try to open it in the kitchen, and out comes the carbon dioxide. That's Henry's law. Because when they were bottling the Coke, they forced the carbon dioxide into the pot at high partial pressure, and then they put the cap on. When you open the cap, you reduce the pressure of the carbon dioxide sitting above the Coke, and when you reduce the partial pressure, Henry's law says that less gas will be absorbed. So when you open the cap of the Coke bottle, you reduce the pressure, and all the carbon dioxide comes out of solution and makes a hell of a mess. So that is an example of Henry's law, and it's important for us in getting gases in and out of the plasma and out of the blood. There's a temperature issue for this too. Have you noticed that if you leave the Coke bottle at room temperature, uncapped, it goes flat very much more quickly than if you leave it in the fridge? So this is affected by temperature, and the gas will come out of solution much more easily at higher temperature than at lower temperature. So the volume of gas absorbed by liquid is proportional to the pressure of the gas. That's what happens when you open the bottle of Coke. And the equation is V equals PK, volume equals pressure times a constant, where K is the solubility coefficient for the gas. So for any gas, all gases have their own solubility. And that's a solubility coefficient, which is a constant in the equation. So carbon dioxide will have a specific solubility, oxygen will have a specific solubility. So those are the gas laws, which are the fundamentals behind what we're going to talk about today and next time. You okay with that? Come across it before, perhaps? So this is how it's relevant. So here you go. The diffusion of gases then depends on a number of different things. Now most of these stay constant. Not all of them, but most of them stay constant. The diffusion of gases depends on the difference in the partial pressure. So the pressure of the gas will determine which direction it diffuses in. It depends on the solubility of the gas. That's going to be a constant for any given gas. It depends on the temperature of the gas. This is that little temperature issue that I asked you to underline in the last slide, and we'll come back to next time. So temperature is an issue. The molecular weight of the gas, clearly that's a constant, and the area of the surface for diffusion. Now normally all of these are constant, except the partial pressure. The temperature is not always constant. I should actually make a note there. Let me just explain to you what I'm getting at with the temperature issue here. And we'll talk about it next time. The temperature in different parts of your body is not always the same. The temperature in working muscles, for example, is higher than the temperature in other tissues. And working muscle is where you need to deliver oxygen. That's the issue about temperature. I'll come back and explain that properly next time. So that's why I keep mentioning temperature as an issue. Because the temperature in working muscle, where you want to deliver the oxygen, is higher than in other places because of the high metabolism going on there. So that's the temperature. Diffusion area might be a problem in some of these diseases that I've talked about. Remember we talked about emphysema, where the walls between the alveoli break down. And so you haven't got a full complement of alveolar walls, you haven't got a full complement of respiratory membrane with which to exchange the gases. So in some diseases, the diffusion area might be an issue. But for most cases, what the main issue is are the differences in partial pressures of the different gases in different places. So let's have a look at partial pressures of gases in different places. First of all, these are approximate partial pressures of the respiratory gases in millimeters of mercury. Let's have a look at the gases that we've got listed here. First of all, in nitrogen. So PN2 is the partial pressure of nitrogen. PO2, partial pressure of oxygen. PCO2, partial pressure of carbon dioxide. And PH2O, the partial pressure of water vapor. So let's just focus on the first row here, atmospheric air. And we can either look at the actual partial pressure, so for nitrogen 597 millimeters of mercury, or we can look at the percentage of nitrogen in the atmospheric air. Let's look at the percentages first off, because it's easier to grasp by looking at the percentage numbers off the top, but they're the same essentially as the partial pressure numbers. So in atmospheric air, the most abundant gas is nitrogen, 78%. But nitrogen doesn't play any part in respiration. It's an inert gas as far as we're concerned. I list it here because it is such a large proportion of the air that we inspire. So 78% of the atmospheric air is nitrogen, but doesn't play any role in respiration. Here are the important two. Partial pressure of oxygen in atmospheric air, about 21%. Partial pressure of carbon dioxide is 0.04%. There's also water vapor in the atmosphere. And this is listed here at 0.5%, but that's going to vary, right? I mean, on a day like this, the moisture content of the air at this temperature is very, very low. So I don't know where it is, but it's almost certainly less than 0.5%. On a rainy day, of course, they're very humid, and the concentration of water vapor is going to be that much higher. We don't have to worry about it. Let's just put 0.5% as a figure. Okay, so that's the air we breathe in. Now, as soon as we breathe it in to the upper respiratory tract between here and here, the only thing that's going to happen, you remember, is that it gets humidified. There's no gas exchange, nothing in the upper respiratory tract, but it is humidified. And so look what happens to the PH2O, goes from 0.5% to 6%. So about 10 times more water vapor in the air once it's in your nasal cavities, throat, and trachea. And thereafter, that doesn't change. So once it's humidified, it's humidified. But look at what the effect of humidification is on the partial pressures of the other gases in the mixture. So here's 6%. So the partial pressure of nitrogen goes down from 597 to 563, and the percentage goes down to 74%. Oxygen goes down from 21% to 20%, and carbon dioxide will go down, but it's much smaller than 2 decimal places here, so it's not doesn't show up at this level of significance. So there is the humidified air, water vapor goes up, everything else goes down consequently. Now then, the humidified air reaches the alveoli. So here we are, the alveoli. Nitrogen doesn't change, nothing's going to happen with nitrogen. Oxygen goes from 20% down to 14%. Where's the oxygen gone? into the blood. The oxygen's gone into the blood. And the carbon dioxide goes up from 0.04 to 5%. So where's that come from? That's come from the blood. So in the alveoli then, the oxygen goes down, and the carbon dioxide goes up, water and nitrogen don't change. Now then, that's the air in the alveoli. What about the air that you expire? Look, nitrogen and water vapor we can forget about now, they don't change. In the expired air, in the alveolar air, 14% of the oxygen. Expired air, 16% oxygen. Why is that? Dead space. So you've got alveolar air, which is depleted of oxygen, and you've got all the dead space air, which still has the same oxygen that it had when it came in. So when you breathe out, you're breathing out a mixture of alveolar air and dead space air, so the exhaled air has more oxygen in it than the alveolar air. Pretty straightforward. And similarly with carbon dioxide, slightly less, from 5% to 4%. So a difference between the alveolar air and the air you expire. And that's due to, that's because it's alveolar air plus dead space air. The double asterisk here, just to remind you that when you give someone mouth-to-mouth resuscitation, your expired air is good. It still has 16% oxygen in it. Doesn't have the same amount of oxygen as atmospheric air, but it still has enough to resuscitate somebody. So your expired air is good for mouth-to-mouth resuscitation if you ever have to do that, and that's why. So that's pretty straightforward. Looks a bit intimidating at first, but not very. You don't need to remember these partial pressure numbers. I would say it's handy to remember the roughly the percentages of gases in different places. But I'm not going to ask you to remember the partial pressures. Again, as with most of these things at this level, the concepts are more important than the actual numbers. Okay with that? So this is the same thing, just focused a little bit more on what we're really interested in. Partial pressure during respiration, atmospheric air, 106, oxygen 160, alveolar air 100, pulmonary veins 100, systemic arteries 100, the cells around about 40, and the veins coming from the cells about 40, pulmonary arteries going through the lungs about 40. So that's the range of oxygen partial pressures that we're going to be talking about in the next little while. So between essentially a low of 40 millimeters of mercury to a high of 100 millimeters of mercury, 40 to 100. And the same thing for carbon dioxide. The alveolar air about 40 millimeters of mercury carbon dioxide, and in the cells and the veins about 46. So that's the range of carbon dioxide partial pressures, and that's the range of oxygen partial pressures. All right. You know what? For some reason, when the schedule came out, they gave me an extra class on respiration. I could have actually, where are we today? Wednesday. I could have actually finished on Friday, but in fact they've given me till Monday to finish respiration. So previously I have to rush this lecture and the next one to fit it. I didn't ask for an extra one, but they gave me an extra one. So it means I can stand here and talk to you like this and not be... So I used to have to combine this one on Fridays almost into one class, which is a bit of a rush. And so what I've ended up with this year is 2 classes, which I can take a bit more easily and not rush the end bit. So that's why I can afford to go slowly for today and Friday. And then the last respiration lecture is on Monday, and that's another story altogether. Just thought you'd like to know that. Okay, so look, we talked about this before, but we can afford to spend time, as I just said, having another look at it. Here is the respiratory membrane. Here it's called the alveolo-capillary membrane. That's okay. Alveolo, the alveolus, capillary membrane. These things often have more than one name. Alveolus, here is the direction of oxygen going across the surfactant, the fluid layer with surfactant on it. So that's where the oxygen will dissolve in that layer there, across the cell, across this interstitial space here, the space between the cells. Interstitial space is space between the cells, and of course it's usually filled with something, collagen and the molecules that are outside the cells, and that's what we call basement membrane in other areas. There, the basement membranes in the interstitial layer here between the cells, through the endothelial cell, and then into the plasma, and then into the red blood cell. And okay, I have something else to say about this, but I'll leave it till the next slide. And then the carbon dioxide going from the red blood cell into the plasma, across the membrane, into the airspace. So these things, remember, this here is plasma. And the gases have to dissolve in the plasma and then go into the red blood cells, and the carbon dioxide out of the red blood cells into the plasma and then across the membrane. So there are two separate compartments on this side. There's inside the red blood cell and there's outside the red blood cell. And that's not always clear from these pictures. I'll come back to that issue in a minute in another picture where I'll ask you to modify it slightly. So the alveolo-capillary membrane then consists of the basement membrane, the alveolar epithelium, basement membranes, and the capillary endothelium. There are no discontinuities in this membrane. The gases have to go right through it. There are no interruptions in it. Gas transport across the barriers by diffusion after dissolving in water. And the barrier is variable in different regions. Even in some of the cartoons, you can see that in some regions it's thicker than others, but it's of the order of 0.1 to 5 micrometers thick. So 5 micrometers, a long way for gases to diffuse. And the gases diffusing across the membrane are affected by a number of different factors. The thickness of the membrane, obviously, as I just said, it's obviously easier to diffuse across 0.1 than it is across 5 micrometers. The surface area of the membrane, we've talked about that. The diffusion coefficient of the gas in water, in other words, gases that diffuse more quickly that dissolve more quickly will diffuse more quickly. The pressure difference on either side of the membrane, which is the most important one, and the rate of blood flow. So that comes to my, my thing. What's that called? The ventilation perfusion ratio. So the ventilation and the perfusion and the rate of blood flow is going to affect the efficiency of the transport. And the exchange needs to occur in less than 1 second as the blood flows through the capillaries. So where they come together, there's gas exchange. And that has to be optimal. And we talked about that last time, the ventilation perfusion ratio. So the gas exchange in the alveoli, the efficiency of it, is dependent on an efficient ventilation perfusion ratio. So here we are looking at the gas exchange, in this case of oxygen. So here are the alveoli, and the gas is coming in, and the PO2 of oxygen is about 100 millimeters of mercury. The blood is coming in here, deoxygenated at about 40 millimeters of mercury, and the oxygen then diffuses down its concentration gradient from where it's 100 to where it's 40, and the blood leaves the lungs oxygenated at about the same oxygen partial pressure as there was in the alveoli. So it comes in at about 40 and leaves at about 100. And so the oxygen leaves the alveolar air, goes across the respiratory membrane. Here is the bit that I want you to just make a change. It's not clear here that in fact, of course, the oxygen has to dissolve in the plasma before it goes into the erythrocyte. This shows it going from the alveolar air straight into the erythrocyte. That's very misleading. So what you want to do is make another arrow from here to here and write O2 there, and then another arrow from the O2 there into the red blood cell. That makes it clear that there's oxygen both in the plasma, dissolved in the plasma, and in the red blood cells. And in the red blood cells, it attaches to HB. What's HB? Hemoglobin, which I know you talked about last time, and I'll say a little bit more about in a little while. And the oxygen combines with hemoglobin in the red cells to form oxyhemoglobin. So in fact, oxygen is in two places in the blood. It's in the plasma, and it's in the hemoglobin. Important concept. In the plasma and in the hemoglobin. Okay, let me come back to that. I can adjust the order of these slides. This is skipped one here. So oxygen is transported in combination with hemoglobin, Hb, in the red blood cells in the form of oxyhemoglobin, but it's also dissolved in the plasma. And here's the thing that's not intuitive. You know this word, intuitive? Doesn't initially sound right, but it is. When we talk about the partial pressure of oxygen in the blood, what we're talking about is the dissolved oxygen, not the hemoglobin oxygen. That doesn't sound right, because when you think about oxygen transport in the blood, oh yeah, combines with hemoglobin, transported around, that's the end of the story. But in fact, although most of the oxygen is in the hemoglobin, it's also dissolved in the plasma. And it's the stuff that's dissolved in the plasma that we're talking about when we talk about the partial pressure of oxygen in the blood. So that's what it says here. It is the dissolved oxygen that is responsible for PO2, not the hemoglobin-bound oxygen. So when I'm talking about PO2 in the blood, I'm talking about dissolved oxygen, not hemoglobin-bound oxygen. Yeah? Any questions? All right. So that's oxygen and the factors affecting or issues about it. And now go back one to carbon dioxide. Carbon dioxide is a bit more complicated. And this is why. So here we are. Here we have the air in the alveoli at about 40 millimeters of mercury PCO2. Here we have the blood coming into the lungs. Remember, it's been deoxygenated, the oxygen's gone, it's now carrying carbon dioxide, which it's got to get rid of, it's got to unload. It's coming in at about 46, remember that range, 40 to 46. The carbon dioxide then goes down its concentration gradient, so it leaves the blood and goes into the alveoli and comes out at about the same PCO2 as in the alveoli. So it goes from 46 to 40 because it has done exchange with the gas in the alveoli. So let's now have a look at what is actually going on here, and you can see it's a bit complicated. Have you come across these kinds of reactions before? Yeah? Where? In, where? Chemistry? Biochemistry, maybe. Well, now we're going to have to talk about it in physiology. It's really important. And the issue is here that the carbon dioxide is in solution. Now, when you dissolve carbon dioxide in water, you get an acid. Do you remember what the acid is? Carbonic acid. And the thing about carbonic acid that is that it's what the chemists call a weak acid. In other words, it doesn't sit around in the jar as carbonic acid. It dissociates into bicarbonate ions and hydrogen ions. And because it dissociates into bicarbonate and hydrogen ions, it affects the pH of the blood. So carbonic acid almost doesn't exist. You go into the chemistry lab and you see bottles of acid on the shelf. You see sulfuric acid, you see hydrochloric acid, you see nitric acid. You never see a bottle of carbonic acid. That's because you can't keep it as an acid. It always dissociates into bicarbonate and hydrogen ions. And the bicarbonate and the hydrogen ions affect the pH of the solution. And this comes to the issue that I talked about right at the very beginning, and that is that respiration affects the pH of the blood and body fluids. This is the whole topic of acid-base balance. Acid-base balance, the pH of the blood. pH of the blood is maintained within very narrow limits, about 7.4. You stray outside those limits, and you develop a condition called acidosis, to acid blood or fluids, or alkalosis, to a high pH of the blood, and that leads to all kinds of problems. And you can see that by dissolving carbon dioxide in the blood, you're forming carbonic acid, you're forming bicarbonate ions and hydrogen ions, which is going to affect the pH of the blood. So respiration affects acid-base balance. Now next week I'm going to start talking about the kidney. The kidney also affects acid-base balance. And between respiration and the kidney, so air in and out, pee in and out, between those two processes you control the pH of your blood. And that topic of acid-base balance, then at the end of kidney, we have to put all that together and address this issue of acid-base balance. How do you control it? And the lucky thing for you is, I'm not doing it. Because it's a rat's nest, this stuff. Acid-base balance, and I mean, I do it. But I don't do it as well as the guy that's going to come in and do it with you. That's Dr. Alexander, who is up in physiology and deals with patients who are having trouble with acidosis and alkalosis and stuff. And he's going to give you 2 classes. Instead of my half a class, which I just scraped the surface, he's going to give you 2 classes on acid-base balance, which is a really tricky subject, and it integrates respiratory and renal physiology. So the doctors in the hospital that deal with patients who have acidosis and alkalosis are often kidney doctors or pulmonary doctors, and they specialize in this side issue, which is not such a side issue, very important issue, of acid-base balance. Okay, so that's why it's important. So it's not just, so the transport of carbon dioxide is not just an issue of getting carbon dioxide from the lungs or from the tissues to the lungs, it's also an issue of controlling the pH in the blood. So here we are, look. What are we going to do now? We're going to exchange carbon dioxide from the blood here in the red blood cells and move it into the alveolar air. Well, the carbon dioxide is carried in the red blood cells and in the plasma, largely as bicarbonate ions. One of the big messages today. Carbon dioxide is carried around the body largely as bicarbonate ions. And so here is the bicarbonate. Look, we put a big word, put the word start here with an arrow to this bicarbonate ion. Here's a good place to start understanding this. So here is bicarbonate ions in the plasma, and this is a transport protein in the membrane of the red blood cell. Remember transport proteins? And this transport protein is a bicarbonate transporter. And it moves the bicarbonate from the plasma into the red blood cell, from there to there. That combines temporarily with hydrogen, which has come from hemoglobin, to form H2CO3. What's H2CO3? Carbonic acid. So there is bicarbonate plus hydrogen, it goes to carbonic acid. But the carbonic acid doesn't hang around. It doesn't hang around because it's a weak acid, and just to be sure, there's an enzyme in red cells called Ca, or carbonic anhydrase, which even more rapidly breaks down the carbonic acid into water, that's okay, that just hangs around, and carbon dioxide. And carbon dioxide, when it is in gas, diffuses very quickly. So it goes out of the red cells, into the plasma, across the cells, and into the alveolar arch, very fast. So that is the equation for how bicarbonate becomes carbon dioxide, and then the carbon dioxide diffuses. Now, it turns out that carbonic anhydrase, that this equation is so important in cells, that carbonic anhydrase turns out to be one of the most abundant proteins that we have. If you look at cells from any of our tissues, and you analyze what the proteins are in those cells, carbonic anhydrase turns up everywhere. So that's an indication of how important this equation is for controlling bicarbonate, carbon dioxide, and pH of the blood. Okay, so that's it then. The bicarbonate ions pumped in, combines with hydrogen, carbonic acid very quickly dissociates under the influence of carbonic anhydrase to CO2 and water, and the CO2 diffuses very rapidly. Just before I leave this picture, let me show you one more thing on it that I haven't mentioned, and that's this. It turns out that CO2 actually combines with hemoglobin a little bit. That might be new. Again, that's not intuitive, is it? You think hemoglobin, oxygen, carry it around. But actually, there's a bit of carbon dioxide attached to the hemoglobin too. And that has an interesting effect. So that's, I think that's going to come up in the next picture or two. I can't remember when it comes up. So this is going to come up again. It's going to come up again in the next class, and it's going to come up again when we do kidney. So this is going to become quite familiar to you, but it is important. Are there any questions that I can address before we just take this a little bit further today? Yeah.
So does that picture hemoglobin also carries hydrogen ions too?
Yes. The hydrogen ions, the hemoglobin can be reduced or oxygenated, and when it's reduced, it can give up hydrogen ions as well. Yeah. So that's how the hydrogen ions are made available by coming off the hemoglobin. Okay, so that was the oxygen. Here are the statements about the gas transport in the blood of carbon dioxide. So what I've said, and what this says is, now think about oxygen. Go back, just cast your mind back to oxygen. Oxygen transported in two places, on hemoglobin, dissolved in the plasma. Think about what I said about carbon dioxide. Carbon dioxide is in 3 places, and here they are. Most of it is carried in the blood as bicarbonate ions. Way the most. I'll show you how much in a minute. Way the most is bicarbonate ions. Very important also in controlling the pH of the blood. It's also carried as bicarbonate in the plasma and the red blood cells. So there's 2 different places that it's carried. In bicarbonate in the plasma, bicarbonate in the red blood cells. Some carbon dioxide is carried in the RBCs in combination with hemoglobin. That's what I just said, yeah. As a compound called carbaminohemoglobin. And it's responsible for an effect, which I think we're going to talk about next time. I don't think we'll get onto it today, called the carbamino effect. So oxygen in 2 places, in the plasma, on the hemoglobin. Carbon dioxide in 3 places. On the plasma, in the red blood, in the plasma as bicarbonate, in the red blood cells as bicarbonate, and in the red blood cells as carbaminohemoglobin. Okay? 3 places. All right, the carbamino effect comes up right now. Let me just see how we do it. Okay, I'm taking my time so much here that we're going to run out of time. All right, look. Here's why the carbamino effect is so important. Here is hemoglobin. Combines to oxygen. I'm going to review that in the next few slides. I know you've already done it. Here's carbon dioxide. Carbon dioxide comes along and says, oh, hemoglobin. I can attach to that too. Attaches to the hemoglobin, and the hemoglobin releases its oxygen. So the attachment of carbon dioxide to the hemoglobin via the carbamino effect causes the hemoglobin to release its oxygen. Can you hold that on the back burner till next time? It's called the carbamino effect. I'll remind you about this and why it's important next time. So that's what this says. As the hemoglobin, hemoglobin concentration increases, the affinity of hemoglobin for oxygen decreases because the binding of carbon dioxide causes a change in the hemoglobin structure that prevents oxygen from binding it. That's my little thing there. Hold that idea until next time. Okay, let's have a quick look at hemoglobin again before we quit today. I know you've done this before. I hope you remember it. Hemoglobin consists of 4 peptides, 4 globin polypeptides, and there are several different types of chains. Commonly, we have 2 alpha chains and 2 beta chains. Each of these polypeptides has a special chemical group in it called a heme group. A heme group. And here is the heme group here. And you can see there it's attached to the polypeptide. Now we're focused on that heme group. Each heme group has a ferrous iron atom. And each ferrous iron atom binds 1 molecule of oxygen. So a molecule of hemoglobin can combine with 4 molecules of oxygen. Fully oxygenated blood contains about 20 mils of oxygen per 100 mils of blood. Don't expect you to remember that, but that's what it is. Okay, so this molecule here of hemoglobin, 1, 2, 3, 4 globins, 4 hemes, can bind to 4 oxygen molecules. So here is our red blood cell full of hemoglobin molecules. And when the blood is fully oxygenated, here, the arterial blood, look, fully oxygenated, the PO2 is 100 millimeters of mercury, and that is about as oxygenated as it will get. And this shows the saturation of hemoglobin in venous and arterial blood. So in arterial blood, here are the hemoglobin molecules. The purple dots are oxygen, and each of the hemes has an oxygen molecule on it. So each hemoglobin molecule is carrying 4 oxygen molecules, except that one. Because when the arterial blood is fully saturated at 100 millimeters of mercury, the hemoglobin is 98.5% saturated. Not quite 100%. That's why that one is missing an oxygen. And the other thing here is that I pointed out and emphasizes that there's also oxygen actually dissolved in the plasma. Or in this case, in the cytoplasm of the erythrocyte, not on the hemoglobin. So this is the situation in saturated blood. In venous blood, where the PO2 is now, you remember, 40 millimeters of mercury, in that case the hemoglobin is only 75% saturated. So even in venous blood, not all of the oxygen comes off the hemoglobin. Still 75% of it left on there. The hemoglobin loses about 25% of its oxygen in venous blood here. And you'll notice that 75% saturated means that all of the hemoglobins are 75% saturated. It's not that 75% of them are 100% saturated and 25% have none. Do you understand what I mean there? They're equally desaturated. So that's the situation with hemoglobin saturation, 98.5% saturated in arterial blood and 75% saturated in venous blood. But remember that it is the dissolved oxygen that's responsible for the PO2, not this. So if this is plasma out here, then there's dissolved oxygen out here, and that is what we mean by PO2. Last slide today. Last slide, just give it your attention. Here we go. Transport of oxygen in the blood. The binding of oxygen to hemoglobin in the lungs is rapid at normal alveolar PO2. The hemoglobin picks up the oxygen very rapidly. And the release of oxygen in the tissues, so you pick it up in the lungs, circulate it to the tissues, and then you give it up. And it depends on different conditions. And it varies with PO2, pH, temperature, and you show this, the release of oxygen from hemoglobin. Here's our hemoglobin. In the tissues, you want the oxygen now, so the hemoglobin gives up its oxygen. That is what we're going to talk about next time, and you express that as what's called a dissociation curve.